soon as I see that. Great. Hi, welcome to RBCM at Home. My name is Kim Goff and I'm a learning program developer at the Royal BC Museum. I'm coming to you from my home on the territories of the Lekwungen-speaking people in Esquimalt, British Columbia. In this series, we're talking with staff of the museum who've been asked to work at home about their work and research. June 7th to 14th is Oceans Week in Victoria. Originally a one-day event on World Oceans Day, this year it is a week-long series of presentations and activities provided by partners around the city to support ocean-inspired events on southern Vancouver Island. Our current situation means that many of these programs are being live-streamed. This week, all of our at-home series will be Ocean Week themed, including a special outside presentation tomorrow. And a good thing it wasn't today, because it is rainy where we are. To kick us off, we have Invertebrate Collection Manager, Heidi Gartner. Heidi, before you tell us more about what your job entails, perhaps we should start by defining invertebrates and give us maybe just a few examples of what's in the collection.
Oh yeah, that's great. So invertebrates are animals without backbones. And the more common ones you may think of are things like crabs and snails and octopuses.
Excellent, great. And today you're going to talk, so you're a collection manager at the museum, you're also a researcher, and you're going to talk a little bit about what that job looks like. What do you do on a day-to-day basis, a bit about what the collection looks like at the Royal BC Museum. Anything else that I'm missing?
No, that sounds great, yeah.
Perfect, so I think you're going to share your screen and give us some pictures and insight to behind the scenes.
Yeah. So hopefully everyone can see that now, but yeah, my name's Heidi and I'm the Invertebrate Collection Manager and Researcher at the Royal BC Museum. So what does that mean? I get to work with a really cool diversity of animals. I get to go to some very amazing places in the ocean and see some beautiful things. And I get to talk to lots of neat people like yourselves. So I'm just going to share an insight kind of what my job is and how I get to play in the ocean as part of my job. So feel free to ask questions along the way, but I'll just kind of take you on part of that journey. And we started off by saying, you know, what is an invertebrate? So some of the classic examples are crabs, sea stars, snails, but there's an incredible biodiversity that we have, especially here in BC, but in the invertebrate world as well. And just to give you a very brief oversight, there's things like sponges, and even within the sponge realm, there's a whole diversity of what they are. They can be minuscule, tiny little things up to kilometers long, big reef-like structures in BC that are over 9,000 years old. We have jellyfish and its relatives. So up here you're seeing hydroids, corals, anemones. I also study weird things called moss animals or brine zones. And if you were to take a microscope and look at the surface of this animal, you'd see all these tiny little structures that are plucking out food from the water column. So again, just incredible weird diversity.
I'm a prairie girl, Heidi, and I have to say it blew my mind when I moved to the coast, and I won't say how old I was, but when I moved here and I realized those are animals because they looked like plants to me. And I just had no experience with them and assumed they were plants.
No, it's very cool. Same sort of thing. And even some people think they're weird rock deposits sometimes, like mineral deposits. But if you look close, they have structure and, oh, it's fascinating. Yeah. Thanks, Kim. With the crabs, there's other arthropods or things with legs. So if you think about your shrimp, your barnacles, your isopods, also the pill bugs that people see when you're on land, those are closely related. And then even within the echinoderms or the spiny skins, the sea stars we discussed, there's also urchins, which I don't have pictured here, brittle stars, sea cucumbers. And with the mollusks, we have along with the snails, we have octopods and squids, chitons, bivalves. Don't even get me started on the worms. Things that look like worms don't even all belong in the same phylum. And then I actually also study a group called tunicates, which are, out of everything on the page, this spiky little weird ball is the thing that's most closely related to us, which is very strange. The majority of my job does happen though at the Royal BC Museum. So you think about these diversity of animals along our coast, it's the museum's job to document BC's biodiversity over space and time. And so what that means is we do have a collection of specimens of these species in the museum. We store them as either wet or dry material, or we even have also a tissue collection. So now I take little snippings of animals when we're out, and they go in really strong alcohol and into deep freezers, and that's how they do a lot of the genetic work that's going on more and more in the modern science age. But each thing, think about it almost like a book. So each specimen has data associated with it, where it was collected and when and by who and what name it's had and all that sort of fun stuff. And so my job is really being a librarian of dead animals in jars and boxes. So researchers will use the data, they'll look at the animals, they want to pull it out from the library and look at it, or the collection. We do send them on loans. So a large part of my job is doing this, keeping this world organized and sharing this data and helping move science along on that sort of use with the specimens. I am also a researcher, and research also does go on in the museum. So this is a very messy picture of our lab. We have microscopes and fume hoods and all those fun stuff. And I study things called taxonomy and systematics, which is, naming animals and how they're all related. But really the fun stuff is I do get to go out and do field research. And so I got my start by studying invasive species or introduced species to a coast. And essentially fieldwork for me will look like going to an area that's kind of human mediated, has anthropogenic structures. So like marinas and docks and aquaculture sites. And I'll just look at the animals that are growing on these structures. So if you have kids and you want to entertain yourself, go to a marina and look for like the dirty, bumpy boys or the old floats that have been stuck in the water for a while, and just turn them over and you'll see incredible diversity of life. And what I was looking for was species that aren't native to our coast that would be coming in and living in these communities and how they would spread from there. But my absolute favorite is we partner as the museum with institutions like Fisheries and Oceans Canada. And this is usually my favorite week of the year. I'll go out for anywhere from 5 days to 3 weeks. And we live and work aboard this vessel. We're usually accomplishing whatever Fisheries and Oceans needs to do for their research. So the programs I typically partner with are related to, again, introduced species, or like species at risk, or we've been partnering with the groups that look at, important habitats and marine protected areas for species along our coast. And this can be in response to, focal species or things like oil spills. But we get to live on this vessel. So this is what the bridge looks like. We'll travel along the coastline. Usually we're going to more remote places. Obviously we wouldn't need a vessel if we were just studying Victoria. So it's often more like the central coast or the north coast of BC. And then it's pretty neat. You sleep in bunk beds. We eat on the board. I have to admit there's a cook taking care of us for all our meals. It's very lovely. And then there's research labs on the vessel. So we have our field gear for when we go out, but we also have microscopes and imaging stations and when we can do our lab components of our work. So this is where I'm usually working late at night after we've been out during the day. And what we do is we typically launch a boat off of the main vessel to go around and do more exploratory work on like beaches and that sort of thing. So we're not doing the large tows. And I'll do anything from crab trapping for different species, where we collect and we document, size and shape and sex and how old we think they are and the different species, to we do kind of intertidal beach work. And this can be everything from just timed walks looking for introduced species to really detailed research where you take a quadrat, which is basically like a square, you mark them out in according to scientific design, and you're measuring and counting and looking at everything that falls within that square. So just very varying degrees of work. And yes, we're doing this in all weather. So it's beautiful and wonderful and hard and cold and challenging at times, but it's the dream for a marine biologist. And I get to see really cool animals. So often we're going out in the spring during the really good tide series. This also happens to be when resources are starting to become really abundant. So a lot of invertebrates time their reproduction with that. So what you're seeing in this image are these snails have laid all these little egg cases here along this rock. Here's another mating frenzy, basically. These are dogwinkle snails, and all those little yellow things there you see are egg capsules, and inside each there's quite a few little tiny eggs, and usually 4 to 8 individuals will emerge at the end of the season.
Per capsule? 4
Yeah,
to 8 individuals per capsule?
Per capsule, yeah. And this is a dark image, but it's really cool because most sea stars don't brood their babies. They don't carry them around, but this species does. And what you're seeing, if you look really close at all these little white dots, are tiny little baby sea stars that it's brooded and are just finally leaving their mama and going out into the world. So some pretty neat things when you stop and take the time and look. I also get to just see beautiful animals in some pretty remote places. This is abalone. It's a species that we have here on the coast that's at risk, actually. And before I started doing this field work, I never actually thought I'd see for sure the specimens in the wild. They've had a really hard time recovering. They were hit really, really hard, and for multiple factors, they're having a difficult time recovering along our coast. So being able to find populations of this are incredible. And you might recognize it more like this. So it was really highly valued for not only its food. Usually there's a big muscular foot here that's really delicious for eating if you like snails and that sort of thing. It's basically like a really big, large, fleshy foot. But also the shell is so beautiful, very important, gorgeous. Used to be very prominent in like indigenous cultures for trade and that sort of thing.
Heidi, before we get too far from those brooding images,
Yeah.
Julia, who's watching with us here on Zoom, was wondering how many of those snails or sea stars make it into adulthood. So if there's 4 individuals from little capsules, how many of those will make it to adulthood?
Yeah, that's a really great question. For a lot of invertebrates, it's such a small percentage of what actually makes it into adulthood. So most sea stars, for example, do this thing called broadcast spawning, where they just take their gametes, they just take their eggs and their sperm, and through environmental cues, usually tide and temperatures and that sort of thing, they just throw their gametes into the water, hope they mix, hope they survive being eaten by the fish, and hope that someday they settle out and grow into adults. So things like that, they have less than 5%. There's indications that there's more success for animals like the ones that brood those egg capsules. But yeah, for invertebrates, there's a lot of energy cost that goes into it to produce lots, so that, because so few actually make it to adult. So.
And Meredith's wondering, how are sea stars born? Were those egg capsules as well?
No, so most sea stars do the broadcast spawning that I just described, where they literally just throw everything in the sea and hope for the best. But the image that I showed before of this one, it's one of the few species that actually broods. So the mother will collect sperm from the water column and she'll fertilize her eggs, and then she just broods them underneath her body until they get to about this side. And then these are the crawl away juveniles that they have there.
And what kind of sea star did you say that was?
This is one of the 6-armed sea stars.
Thank you.
Mm-hmm. Any more good reproduction questions? I can actually say that invertebrate reproduction could be its own talk. They're fascinating adaptations to living in this marine world, everything from asexual reproduction to hermaphroditism, which is really common, but also hermaphroditism where you begin your life as a male and at a certain age you turn to a female. And then the courting behaviors in octopods. So yeah, we can set up another one.
Heidi, let's do it for next Valentine's Day.
Sounds good. Yeah. So northern abalone, and then this was pretty neat. Just again, weird things that I get to discover and play with on the shore is this was, I lifted up a piece of bark and found hundreds of these little individual snails underneath here, what look like snails, but when you look closely, they're all hermit crabs. And so I still to this day do not know why they were all there. It could have been the temperature, it could have been a mating season, it could have been all these other factors, but pretty neat that we got examples of this that we were able to show. And then here's another beautiful, weird insight into the invertebrate world and research that we do is we're looking at all the animals that we can find on these beaches. And this is actually a worm. So these white tubes that you sometimes see on rocks, these are called serpulid worms. And what they do is they stick the top heads of their body, have been adapted to have all these little feeding tentacles, basically. They kind of look like feathers, and they grab food from the water beside them. And then if you can see here, there's like a hard circular structure kind of hiding amongst those tentacles. And what happens when they're disturbed, they pull everything in, and then they can close off the top of their worm tube. And so one of the things that's neat is that the different species actually have different shapes of this thing called an operculum that blocks the top of their tube. So that's things that I can help with as an invertebrate taxonomist, is I can tell them what species it is based on the shape of this operculum plug. And then documenting invasive species. This is a beach we went to, and all of these snails here are an introduced species and just covered this mudflat. So thinking about how that changes the ecosystem and what it's doing. And particularly if you notice, this is eelgrass. Eelgrass is a really important habitat in BC. It does everything from carbon sequestration to nurseries for important fish species. So documenting these changes in BC are really important. An example of some of the diversity we find, this was just for one of these trips we joined with Fisheries and Oceans Canada. We took the time, we took images of everything live, and these were all the diversity of just the sea slugs or the nudibranchs that we found during that one trip. So BC has some incredible biodiversity that not very many people know about until you start exploring. When we do these research partnerships with Fisheries and Oceans Canada or other institutions, we're usually trying to help them accomplish their goals. And in exchange, they let us collect specimens for the museum when we're able. And what we do is we bring them back to the museum. And this is an example of one of my volunteers helped me process. So they've now been properly relaxed and preserved, put in appropriate jars. They now are getting the labels, and then they'll get shelved in that system that you saw before. Why we're doing it, again, sometimes we know something exists in BC, but we didn't have an example in the collection, and that's really important for researchers to be able to have morphological samples to examine. We document things like range extensions, and also sometimes we find new discoveries. So for example, we didn't know that this species actually existed as far north as British Columbia. So this is a new record for BC, and we're sharing this again with the scientific world through our online records or through publications or through partnering with certain institutions. And just for example, this crab that I was talking about, this is an introduced species to our coast. It's the European green crab. And we've been working with Fisheries and Oceans Canada, and they've been documenting the spread. So it was originally introduced to the west coast of Vancouver Island. And over the years, it's been slowly spreading south and north. And so we're documenting that spread and doing some ecological investigations along the way of what they're doing to the certain habitats. And also taking, like I told you, those tissue samples. So now tissue sampling is a really important part of research. And so you basically can DNA sequence or barcode the biodiversity of life. And so when you look at the DNA, it basically makes up, just think about it almost like those barcodes that are on your like food products and how it identifies a unique thing. That can actually identify unique species. And so we're contributing to that research that largely goes on the east coast of Canada at the Biodiversity Institute of Ontario, but to numerous programs as well.
Heidi, how do you keep that DNA?
What we do is we take little tissue samples, and they're usually, they can sequence from like tissue that's just barely bigger than a period. So we just have to take a little bit and we put it in a tiny little jar, and we put it in the strongest alcohol we can, 95% ethanol, and then we store it, and we have freezers that are minus 80 degrees Celsius at the museum. So super cold. Mm-hmm. Mm-hmm. And that slows down any sort of degradation that should happen over time. As cool perks of my job and studying the ocean, I get to see some beautiful areas on the coast. And I just wanted to share these briefly, just so that I know not everyone has the opportunities that I have to go there. And, you know, we protect what we love, and I want everybody to love the remote coast. So we see some incredible things when we're boating around. All wildlife, you know, on our coast really does interact with the ocean. For Oceans Week, we got to think beyond just submerged. There's wolf here on the Central Coast. So cute. And then I have to admit, I've seen some glorious displays of whales as well. I know I'm invert biased, but yeah, it's pretty spectacular when you see whales jumping. And when we are working with places like Fisheries and Oceans Canada, they will stop. They take out their really good cameras with the good lenses, and they usually take photos and try and share that with their marine mammal colleagues who can identify whales based on, you know, the fluke patterns and shapes and those sorts of things. So it's beautiful to stop and watch, but we are contributing to science, which is fun. And I was super lucky. I got to see one of the rarest sights in BC, which is a basking shark. So basking sharks are sharks that have been nearly extirpated from our coast. There's so few left, and they used to be really abundant. So what you see in this image is it's like this big, large, scary mouth. It's not actually scary at all. This is a plankton feeder. So they actually don't even have teeth, and they swim through the ocean. And what they do is they're filtering all those little tiny plankton bits from the water in their gills. And so they swim back and forth across the surface of the waters, skimming out plankton. And they used to be so abundant that fishing vessels and boats used to find them an annoyance. And they even put slicers on the front of the boats a long time ago to decrease the numbers of pests bugging their fishing vessels. So we now have next to no basking sharks on our coast. And I have to say it was pretty exciting to see it. And it was in an area they weren't really expecting it. And this is what it looked like from the boat that we were on. It just had this, you know, the shark fin, and it's got that little nose. And when we were kind of at the right angle, you could see that it was doing its basking, it basks in the sun, and it does the plankton feeding back and forth. So it was really beautiful to see. And just to warn you, we do have to maintain that distance when you're observing marine wildlife. Please be safe with that, and just know that these images are taken with, you know, the good camera, the good lens, and then we're cropped. So let's always respect our marine wildlife.
Thank you, Heidi. Just recently, there are new management measures that just came out for 2020. So if you're someone who spends time on the water, goes boating, or even canoeing or kayaking, it's a new 400-meter distance from any marine mammal like a whale, or in this case a basking shark, and scientific vessels, and some of the whale watching boats will have a special ability to go up to 200 meters. But all those measurements are online, so if you're someone who goes on the water, please make sure you check those out.
Yes, thanks, Kim. This was actually a couple years ago, so it is quite a bit further now. I think they're encouraging that you no longer are even in this yellow zone.
That's right, yeah.
Even for kayakers, you maybe should be careful. There's been a few videos of whales breaching on kayaks lately. So the last part of my job, I'm a librarian. I get to go out and do research. It's fantastic. But the other fun part is communicating science. So I do get to talk at the museum. We interact with the public whenever we can. And then we get to talk with people here through this sort of platform. And I was going to finish there, but I'm just going to show you really quickly. I told you that I study two specific kind of taxa group, those weird and strange ones. I'm just going to show you very quickly. This is what I was talking about. So those brine zones, this is another example of like a big colony that can grow on rocks. This one's kind of made more of like a mushroom shape. But when you put them under the microscope, this is what the surface of them look like. Aren't they beautiful? And then out of these like big holes come those feeding kind of plucking structures called lophophores. And then tunicates is another group I study, and they're basically filter feeding sacs. They take water in, they filter food out of this, in this big structure called a pharynx, and they shoot the waste out the other end. And they can be solitary individuals, or they can be colonial, where you have lots of little individuals sharing the same space. And again, just beautiful animals that are super strange until you start paying attention to them. And this is kind of what the biodiversity of them looks like. And the reason that I said that they're most closely related to us is that they are, when they are juveniles, they share the chordate characteristics. So they have a dorsal hollow nerve cord, a notochord, pharynx with gills, but they lose it when they become adults. So very strange animals, closely related to us. And again, this is how I kind of got my way in, because they can be pests when they become introduced, and particularly for aquaculture. They've cost millions of dollars on the east coast of Canada, and yeah.
Because they form on the nets, or why are they such a nuisance?
They're both, really. So what you're seeing in this image is these are like cages for aquaculture that are now completely covered in invasive tunicate species. So they kind of like to grow over things. Again, this is the same thing on this image here. This is a solitary species, but on a rope. And then I've showed close-ups of, so not only do they increase like the handling time of the machinery and gear, but they can actually have direct stock loss as well. So here's an oyster completely covered in a species and mussels covered in species. So it's kind of a double whammy when they take off.
And you said they're invasive, so how are they getting here?
Interestingly enough, they are being introduced to our coast usually by human-mediated factors. So they can grow on things like ship hulls. But the way that we started the oyster aquaculture on our coast way back when, we've been doing oyster aquaculture for a long time, is our main oyster that you see and that you eat is actually from the other side of the Pacific. And what they used to do is they basically used to go to like Japan, scrape up all the oysters off the beach, bring it here and dump it. And so there's been over something like 70 or 80 species that have been traced back to the oyster aquaculture industry that have been introduced to the Pacific coast of North America.
Oh, wow.
So aquaculture is kind of the big one. And then now these animals are becoming so much more common in our worlds and around sites like, for example, if they come in on big boat hulls, they can get into marinas and that sort of thing, and then they grow on recreational boats as well. So if you ever find weird tunicates on your boat, just know that cleaning your boat before you travel to other areas is always a very, very, very good idea.
Absolutely.
Yeah.
Heidi, there are a couple of questions here.
Yeah.
One is from Riley, who's in grade 5. And Riley is curious about very young sea stars. What do they look like? I don't know if you could have a photo or if there's something online that you know of that we could share, but could you maybe even just describe what a very young sea star looks like?
Yeah. So they are basically beautiful little baby tiny stars. So echinoderms get their, that's the name of the phylum, and that's it translates from Latin into spiny skin. And so the juveniles often look similar to the adults, but they don't usually have nearly as much color. And all the little spiky bits on them look more pronounced. So if you get a juvenile sea star under a microscope, it almost reminds me of a snowflake, because it's a lot more spiky than the adults, or it kind of has that appearance. So when I showed a brooding one earlier, noticed that the adult was kind of dark colors. They were more white, and their skin hadn't really, it kind of looked spiky, and it hadn't really taken on the color yet. Yeah.
That's a beautiful description. I'm imagining little, yeah, almost like, as you said, snowflake kind of things. And yeah, that sounds neat.
But what's really neat is when they're not juveniles, when they're in the water column, when they're those larvae, they're called, they look completely different. And that's the same way with a lot of invertebrates. So if that's another thing, is sometimes it's fun pairing up the larvae with the adults and just being like, whoa, this is what that becomes. So they do have long arms in the larvae, which is neat. So I hope that answers your question. Definitely feel free to Google sea star babies.
Oh, cool. Okay. Well, this is sort of related to the life. This is related to the life cycle. This is from Lori, who's watching on Facebook. Hi, Lori. What do dog winkle snails look like when they come out of their eggs, and how long does it take them to get their shells?
So the ones that I showed in the presentation were in those like yellow egg capsules. What happens in there is really fascinating and kind of gross. They start off looking basically like little yellow balls. And actually each egg capsule usually has something more like 10 to 20. And over time they eat each other and only 4 to 8 usually emerge. And when they come out of those capsules, they look like really tiny versions of the adult. So they already have their shell, and it's actually something called the protoconch, and this may be getting too much, but if you look at the very tops of snails, you ever find a seashell on the beach, that you can usually see at the very, very tip, there's a tiny little section that you can almost see a line demarcation. And that's usually showing where the juvenile was, and then where it then became an adult and started growing its adult shell. So.
Oh, neat. Any particular species of on dogwinkle snails you'd see that, and other snail shells as well?
I'm trying to think which ones it's easiest to see it on. I can't think off the top of my head, but it also depends on kind of the wear and the growing over of the specimen. So look for things that look cleaner and smoother, and you might be able to see it a bit better. Mm-hmm.
Well, that's great. Well, thank you so much, Heidi, for showing us the world of invertebrates, and got a great picture of what your job is like. And I must say, it sounds like a fascinating world of discovery that you have chosen, and feel that, yeah, you do such great research as well as taking care of the collection. So thank you.
Oh, thanks, Kim.
And yes, when next time I go to the beach, I'm going to look for those little marks on the shell, see if I can tell that.
Okay.
Wrapping up now, I just wanted to remind people that the Royal BC Museum is getting ready to reopen on Friday, June the 19th. And as a staff member, I'm thankful for the teams who've been working hard to ensure the safest conditions for our staff, volunteers, partners, and visitors. You can find out more about our reopening on our website. We will be continuing our at-home kids, and outside for the foreseeable future. And links for all of those programs are on the museum's website as well. I'll be back on Thursday with the curator of vertebrate zoology, Dr. Gavin Hankey, and he's going to talk to us about fish, and particularly the fish you might be able to see from the shoreline. And I want to thank you for including us in your school time. I understand there's a grade 4 or 5 class watching us. Thank you very much for joining us. I hope you all get excited about science and about invertebrates in particular. Thanks, Heidi, and thank you, everyone.
Thank you, everyone, and thank you, Kim. Bye, everybody.